everyone, welcome back to my YouTube channel IGCSE Science Academy. Today we are going to explain one of the most important topics of biology for FPSC and other competitive examinations, mammalian gametogenesis and fertilization. In this session, we will cover 30 most important and frequently repeated MCQs for the post of biology TGT in FPSC. Today's topic is one of the most important in biology for FPSC and other competitive exams. So let's get started. First topic from today's session is spermatogenesis. We will cover 6 most important and repeated questions from this topic. First, let's discuss about spermatogenesis. What is spermatogenesis? Spermatogenesis is basically a biological process by which male gametes, sperm cells, are produced in the testis. It takes place in the semiferous tubules of the testis and involves several stages: multiplication, growth, maturation, and transformation. So spermatogenesis in simple words is the process of forming sperm from immature cells in males. Question is the process by which spermatogonia develop into mature spermatozoa is called. It's option B, that is spermatogenesis. Here is the explanation. Spermatogenesis occurs in the semiferous tubules of the testis. By the explanation, you can cover one more MCQs from the relevant topic. Question 2 from the topic is spermatogenesis occurs in which part of the testis? It's the option C, semiferous tubules. Explanation: semiferous tubules are lined by Sertoli cells which support developing sperm. Question is which cells support and nourish developing spermatocytes? It's option B, Sertoli cells. Explanation is Sertoli cells, nerve cells, provide a structural and nutritional support to developing sperm. Next question is duration of human spermatogenesis from a spermatogonia is to mature sperm. It's option C, 64 to 74 days. Explanation is spermatogenesis takes 64 to 74 days in humans. Next question is transformation of spermatids into spermatozoa is called. It's option B, spermatogenesis. Explanation: spermatogenesis involves acrosomes formation, flagellum development, and cytoplasm shedding. Next question is the acrosome of a spermatozoon is derived from. It's option C, Golgi apparatus. Explanation: the acrosome cap over sperm nucleus is derived from the Golgi apparatus. The next topic from today's session is oogenesis. Here we will also cover 6 most important and repeated questions for your competitive exams and FPSC exams. First, let's discuss about oogenesis. Oogenesis is the biological process by which female gametes, ova or egg cells are formed in the ovaries. It occurs in ovaries. What are the main stages? Main stages are multiplication, growth, and maturation. Unlike spermatogenesis, only one functional egg is produced from one primary oocytes. Question from this topic is primary oocytes are arrested at which stage in human females? It's option A, prophase of meiosis 1. Explanation is primary oocytes remain arrested in prophase 1, diplotin stage from fetal life until ovulation. Next question is completion of meiosis 2 in the secondary oocytes occurs only after. It's option C, fertilization by a sperm. Explanation: without fertilization, the secondary oocyte degenerates without completing meiosis 2. Next question is the first polar body is produced at the end of. It's option C, meiosis 1. Explanation: unequal division at the end of meiosis 1 produces a secondary oocyte and a polar body. Question is how many functional eggs are produced from one primary oocyte? It's option D 1. Explanation unequal cytokinesis concentrates cytoplasm in one functional egg. 2 to 3 polar bodies degenerate. Next question is which hormone triggers the LH surge causing ovulation? Option C high estrogen levels. Explanation high estrogen from the dominant follicle causes positive feedback triggering the LH surge. Next question is the zone Peludicea is primarily composed of. It's option B glycoprotein ZP1, ZP2 and ZP3. Explanation ZP3 is the primary sperm receptor for species-specific binding to the egg. Next topic from today's session is fertilization. Here we will cover 8 most important repeated questions. First, let's discuss about fertilization. Fertilization is the process in which a male gamete sperm fuses with a female gamete ovum, egg, or egg to form a zygote. In humans, it usually takes place in the fallopian tube, which is also called oviduct. The key steps of the fertilization are sperm reaches the egg. Once sperm penetrates the egg nuclei of sperm and egg fuses, a diploid zygote 2N is formed. The first question from the fertilization is capacitation of spermatozoa primarily occurs in option C female reproductive tract. Explanation capacitation enables sperm to fertilize the egg, takes 7 hours in humans. Next question is the acrosome reaction in sperm is triggered by option C both progesterone and ZP3. Explanation ZP3 triggers calcium ions in flux which progesterone from cumulus cells facilitates the acrosome reaction. Next question is which enzyme helps sperm penetrates the zona pellucida? It's option C both hyaluronidase and acrosin. Hyaluronidase disperses cumulus matrix. Acrosin, which is also called serine protease, digests the zona pellucida. Next question is the cortical reaction prevents polyspermy by its option B hardening the zona pellucida. So the explanation is cortical granules release zona hardening enzymes, the slow permanent block of polyspermy. Next question is the fast block to polyspermy involves its option C membrane depolarization. Explanation sudden depolarization of egg plasma membrane prevents additional sperm fusion within seconds. Next question is fertilization in humans normally occur in its option C ampulla of uterine tube. Explanation the ampulla widest part of the fallopian tube is the normal site of fertilization. Next question is fusion of male and female pronuclei syngamy forms option C zygote. Explanation syngamy restores the diploid chromosome number to form the zygote. Next question is the sperm contributes which of the following to the zygote? It's option B centriole and chromosomes. Explanation sperm contributes chromosomes and the centriole mitochondria are maternally derived. The next topic from today's session is hormones. Here we will complete 3 most important questions. First, let's discuss about hormones. What are hormones? Hormones are chemical messengers produced by glands in the body that control and coordinate different functions of growth, development, and metabolism. Where they are produced by endocrine glands, ductless glands such as the pituitary, thyroid, pancreas, adrenal glands, etc. How they work: release directly into the blood, travel to target organs, affect specific body function. First question from this topic is, which hormone maintains the corpus luteum in early pregnancy? It's option C, HCG. The abbreviation for HCG, H for human and C for chorionic and G for gonadotropin. So answer is HCG. Explanation: HCG forms the trophoblast, maintains the corpus luteum until the placenta takes over progesterone production. Next question is, testosterone in the testis is produced by, its option C, Leydig cells. Explanation: Leydig cells stimulated by LH are the primary source of testosterone. FSH in males acts primarily on, its option C, Sertoli cells. Explanation: FSH stimulates Sertoli cells to produce androgen-binding protein ABP, concentrating testosterone by spermatogenesis. Next topic from today's session is embryology. Here we will complete 4 most important and repeated questions. First, let's discuss about embryology. What is embryology? It is the branch of biology that studies the formation, development, and growth of an embryo from fertilization to the early stages of development. It includes fertilization, cell division, formation of embryo, development of organs, and body parts. Question is, the process of cleavage produces cells called, option B, blastomeres. Explanation is, cleavage produces blastomeres, progressively smaller cells without overall embryo growth. Next question is, the morula stage consists of approximately how many cells? Option C is the correct option, that is 32. Explanation: the morula mulberry is a solid ball of 32 cells formed after several cleavage divisions. Next question is, implantation of the blastocyst normally occur in the, its option B, posterior wall of the ureter. Explanation: blastocyst implants in the endometrium, posterior uterine wall, around day 6 to 10 post fertilization. Next question is, the inner cell mass embryoblast gives rise to, its option C, the embryo proper. Explanation is, ICM forms the embryo proper, the trophoblast forms the placenta. So the full form for ICM is its inner cell mass. Next topic from today's session and the last topic, that is meiosis. Here we will cover 3 most important questions. So first, let's discuss about meiosis. What is meiosis? Meiosis is a special type of cell division in which a single cell divides twice to produce 4 daughter cells, each having half the number of chromosomes as the parental cell. Where it occurs in reproductive organs: testes in males, ovaries in females. What is the main purpose of meiosis? To produce gametes, sperm and egg cells, to maintain chromosome numbers from one generation to next generation. One diploid cell to N to 4 haploid cells N. Question from this topic is, crossing over during meiosis occurs at which stage? It facetine. The correct option is facetine. Explanation: crossing over at chiasmata during facetine generates genetic variation. Next question is, chromosome number in a secondary spermatocyte is, its option B, 23 haploid. So explanation: after meiosis, one secondary spermatocytes contain 23 chromosomes, each still a double chromatid. Next question is, genetic variation during meiosis is primarily produced by, its option B, crossing over and independent assortment. Explanation: two mechanisms crossing over prophase 1 and independent assortment metaphase 1. The key points to remember from today's lesson. These key points can help you to secure better in your examination. First is spermatogenesis, 64 to 74 days. Semipherous tubules, Sertoli cells nurse sperm. Oogenesis: primary oocyte arrested in prophase 1. Secondary oocyte completes meiosis II only after the fertilization. Fertilization: ampulla of ureterine tube (ZP3) triggers acrosome reaction. Zygote is equal to diploid. Polyspermy block: fast depolarization plus slow cortical zona reaction. Hormones: LH, Leydig cells, testosterone, FSH, Sertoli cells, ABP, HCG maintains corpus luteum. Meiosis: crossing over at fecitine, independent assortment, genetic variation. That's all for today's session. Stay tuned for the next video where we will discuss another important topic with MCQs. Thank you and stay tuned and connected. If you like the video, don't forget to share, subscribe and like.